Okay, so we are ready to go, I think. Except for that thing is off center again. And that should, no, still not there, is it? Okay, that should get us there. All right, so today I'm going to finish talking about characterizing and purifying proteins. I'm not going to say much about purification today. Most of that was yesterday. Today I'll talk a little bit about characterizing proteins, and then I'm going to spend most of the time today talking about hemoglobin. So hemoglobin is a very, very fascinating protein. It's a very important molecule for us to understand. So I want to go through and spend some time talking about that. A couple of you have asked me, go through, I don't click on every single link on here. Does that mean you're responsible for them, et cetera, et cetera? And the answer to that is it's the same as anything else. My guideline is if I don't talk about it, I'm not going to hold you responsible for it. So if I don't talk about something, you know, that's fine. There's things that are there that are there for your information. And I do change things a little bit each term, what I do talk about and what I don't talk about, which is why I don't recommend studying from old exams, because things do change periodically. Okay. One of the first things I want to talk about today is the actual purification process. Okay. So this may seem like it's an exercise in giving you something to calculate, but in fact it has a practical importance. So when we look at a purification, when I go to purify a protein, you probably didn't think too much about it, but you say, well, you just go and you purify it. Well, I can't just purify a protein unless I know something about it. If I'm trying to purify a protein, for example, I need to know, well, what does the protein do? Does the protein, for example, catalyze a reaction that oxidizes glucose? That might be an example, okay? Well, if I know what that reaction is, then I have a way of measuring during the purification process which fraction my protein is in. If I don't have that, I have no way of detecting what my protein is. So part of the purification process is monitoring what's happening to the samples as you're purifying them. And what you see is that you don't get 100% recovery. That's not surprising. Okay? But that as you go along, you actually see that you darn sure better be getting purer material or you're wasting your time. And so part of what we're looking at here is the issue of how much material of my desired protein am I purifying and how much of undesirable protein am I getting rid of. Okay? Well, this is a sort of a scheme. You can see this person was purifying a protein, and they went through several different methods. 1, 2, ion exchange 3, gel filtration 4, affinity chromatography 5. So they did about 5 different things here. And what you see as you look at these numbers that are on this plot are that their protein got purer, no surprise, and they got rid of an awful lot of junk. So let's just step through and see what they did. So I haven't talked about all these methods. So for example, I haven't talked about salt fractionation. It's just another way, another step in a purification process that a person might have. Let's look at what this person started with. They took cells, they busted them open. That was the homogenation part. And when they did that, they got 15,000 milligrams of protein. And yes, it's very easy to measure how much protein you have in a sample. There are very simple methods for doing that. They took that homogenate, and they took a small aliquot of it out, and they measured to see, well, how much of this reaction this thing would catalyze. So I might define a reaction of a unit as, say, 1 microgram per mil per hour, okay, something like that. So I have a unit definition, and this person's done this assay, and they say, well, there's 150,000 units that I have in this total tube if I measure it out. And when I divide this out, I get a specific activity. And by the way, specific activity refers to units, okay, that you have per milligram of protein. They have 10 units per milligram of protein because they had 150,000 units, and they had 15,000 milligrams of protein. The yield they had was 100%. Well, of course, it's 100% because they haven't done anything yet. They haven't thrown anything away. That's just their starting material. So the starting material, by definition, is going to have 100%. The purification level is 1, and we'll see where that comes into play when we go to the next step. In the next step, they've done salt fractionation, and what we discovered in the salt fractionation step was they lost a bunch of stuff. They got rid of almost 11,000 milligrams of protein, and they only lost a little bit of activity. Okay? So their activity went down from 150,000 to 138,000 units. If they did the math here and they divided 138,000 by the milligrams, they found that they got a specific activity of 30 units per milligram. Okay? Now, the fact that their specific activity went up meant that they had more units per milligram of protein. That meant they got rid of a lot of protein they didn't want, which is what the numbers sort of showed you. And so they've purified this material. They have, in fact, purified it threefold. The threefold being the specific activity, what they have now, compared to what the specific activity was that they started with. 30 divided by 10 gives 3, and so this guy has been purified threefold. How much yield did I have? Well, the yield that they had was how much of the desired protein that they had compared to how much they started with. So the yield was, in fact, 138,000 divided by 150,000 times 100, and they discovered that they had 92% yield. That's pretty cool. 92% of their original protein is there, yet about almost 3/4 of their undesired protein disappeared. So they've done a pretty good purification step there. You can continue through here. You see at each step they are losing more and more protein. So they started with 15,000 units of protein. They got all the way down here to affinity chromatography, and they only ended up with 1.75 milligrams of protein, meaning they got rid of an awful lot of junk. But that still had 52,000 units in it. Okay? That gave them a specific activity of 30,000 units per milligram. Okay? So it went from 10 to 30,000 per milligram. That was the 3,000-fold enhancement that they achieved in going through this process. They got rid of 99% of the stuff they didn't want, and they're left with a relatively pure compound right here. Now, there were other things they would have to do to determine if it was absolutely pure or not, and we're not going to do that here. But suffice it to say that they've gone through a very good purification, and they've retained 35% of their material. You think, well, they lost 2/3 of their material, but they also lost 99.9% of the things that they didn't want. So they did a pretty good job on this purification. Now, I think you should understand what this table tells you. I think you should be able to understand how to do calculations based on this table. Okay? So, are there questions based on the table that I can answer for you here? Yes?
I don't exactly understand. The total activity is arbitrary, and then you decide what activity you're looking for that would be specified by the starting experiment.
That's right. So that's a good question. So the activity the unit definition that I would have would be something I would carry all the way through the experiment, and as long as I was consistent in that unit activity, then I get a meaningful quantitative number here. That's correct. Was that clear? Or are you guys that sleepy? Afternoon classes I find are really tough to kind of keep people's eyes open. I'm not sure. I think they're worse than morning classes in that respect. Okay. Let's see. What was this one? I can't remember what this was. Okay. So this is simply showing you a sort of a very simple way on a gel that might look. Here was the starting material there. After one step, it got better. After a second step, it got better. Next step, it got better. And bang, by the last step that we had there, things were pretty much pure. One of the ways that we would determine if we had material pretty much free of other things would be running a gel. That's why a gel is very useful. If I saw another tiny band up here, I would say, well, there's something else in there I've got to be worried about. But a gel would be a good way of determining, well, how pure my material actually is. It's not an absolute answer, but it gives me a pretty good ballpark about how pure my material is. Okay. Let's see, S values. I will mention them here because I think my highlights, actually I had an S value in one of the highlights if I recall correctly, and I apologize for that. The S values is not something we have to concern ourselves about. In fact, you're not responsible for them, but I'll tell you about them if you ever hear about them so you know what they are. S values derive their name from Svedberg, who used centrifuges as ways of estimating molecular size. And so the unit was named in Svedberg's honor. Basically, the larger the Svedberg value, that is the S value is, the greater the molecular weight. Okay? There's not a linear relationship, though. So simply a larger S value means a larger molecular weight, and that's for our purposes all that we really need to know there. Okay. Well, let's turn our attention now to characterizing proteins a little bit. I'm going to go through this, some of this fairly quickly, but I want to point out a couple things to you. A lot of the things that we used to talk about with respect to proteins, people don't even bother to do anymore because it's so easy to determine protein sequence by simply sequencing the genome as opposed to sequencing individual proteins. It is sometimes necessary to actually determine the sequence of a protein, and there are some techniques for doing that, and I'm just going to very briefly mention them. One of the old ones is wanted to know what the amino acid composition was of a protein. Well, it's pretty unusual you'll have a protein that won't have every amino acid in it. Exceptions might be if you had very small peptides. There are very small peptides that may only have a handful of amino acids. And in those types of analyses, being able to determine the amino acid composition of a small molecule would be useful. These rely on our ability to hydrolyze, that is to break the bonds, break the peptide bonds between amino acids in a given protein. And as I mentioned, I think in response to a question yesterday, one of the ways we can do that is by treating it with HCl with the heat over a span of about 24 hours, and that will generally break all of the amino acids down, or break the peptide down to its constituent amino acids. And then we can use a technique like, in fact, this doesn't even show HPLC, but most commonly we would use HPLC today as a way of separating the amino acids. They would have distinctive patterns where they would elute from the column. And we could say, oh, well, this peak that came out at 2 minutes after injection is aspartate because I know that's where aspartate comes out. I can put aspartate in there, and that's where it elutes. So I must have aspartate in this peptide. I've got glycine in this peptide. But look, I've got twice as much glycine as I have aspartate. Maybe I've got 2 glycines and 1 aspartate, something like that. Okay. So pretty straightforward analysis that I would do for amino acids. There are fluorescent labels that we can put onto amino acids, and the reasons we put those fluorescent labels on there is because amino acids themselves don't absorb light for the most part very readily. Tryptophan being one exception to that, but for the most part, most amino acids aren't very good at absorbing light. So if we want to detect them coming out of a column, we usually have to derivatize them in some way by adding a constituent to them. Ninhydrin is one of those constituents, and that's just sort of a techie thing for you to consider, fluoresamine being another. I'm not going to talk about Edmond degradation because Edmond degradation is an ancient method of determining the sequence of amino acids in a protein. But I do want to mention it with respect to how advanced our technology has become. So in the old days, meaning probably prior to about 1985, if a person worked in a laboratory and they isolated a new protein, they really wanted to and they needed to know the sequence of amino acids that comprised that protein. And at that time, about the only method that was available was this chemical degradation method called Edmond degradation, in which a series of chemical reactions sequentially cleaved off amino acids from the either the amino or the carboxyl terminus. I believe it's the amino terminus. And as they were taken off of the protein, they were analyzed by, say, HPLC or some technique like that. So if you're very careful and you take them off one at a time, you can actually analyze each one and ultimately determine usually a good deal of the sequence, not all the sequence. But the process was exceedingly time intensive. So to give you an idea, to determine the 10 amino acids at the amino terminus of a protein by the Edmond degradation method might well take over 24 hours. Might take well over 24 hours. All right. Now, Today, with DNA sequencing technology, we can sequence the DNA that codes for those proteins at a phenomenal rate. We can sequence entire bacterial genomes. It's been done in a single day. A single bacterium might have 5,000 or 6,000 proteins. Okay? That would not just be the part of the amino acid sequence of one end, but that would be the sequence of every protein in that entire organism. In a sophisticated lab, they could do that in one day. So, not surprisingly, people don't do much in the way of Edmond degradation and protein sequencing unless they absolutely have to, and they've got a lot of time on their hands, which is not something that people tend to have anymore. There are some reagents, however, that we need to be aware of that have been used traditionally for analysis of proteins, and they're still used today. Now, one of the methods that we use today, I'm going to talk about it in just a little bit, it's called MALDI-TOF. And MALDI-TOF is a way of determining molecular weight of materials, and the molecular weights of those materials can be linked to the weights of amino acids in a protein, and if we do it carefully, we can actually get the sequence of amino acids in a protein by looking at the pattern of molecular weights that we have in that protein. We can't do MALDI-TOF on entire proteins, though. Okay? They're too big for accurate enough analysis. So we have to work on subsections of proteins. And to make subsections of proteins, we have to have enzymes or chemical reagents that will break the protein into smaller, consistent pieces, meaning we need to have enzymes or reagents that cut the protein at specific places. So that's what I'm getting ready to tell you about here. The first is a reagent, that is, it's a chemical that cuts a protein consistently at a very specific place. This reagent is called cyanogen bromide. And cyanogen bromide is very useful for breaking proteins into smaller pieces because wherever it encounters a methionine, it breaks the polypeptide backbone. So the peptide we see, here's the polypeptide backbone, there's our peptide bond, and we see that this guy, where the peptide bond was, gets an unusual derivatization that happens over here next to the methionine. So wherever there's methionine, we basically break the polypeptide chain. The beauty of this is it's consistent. We only break at methionines. And we have to have consistency. Otherwise, if we were randomly cutting things, then we wouldn't know how to put the pieces together. So cyanogen bromide is a very useful reagent for breaking peptide or polypeptides at a given place. It is the only chemical reagent that I will talk about here. There are several enzymes that work similarly, and we're going to talk about one of them here, and there are others that we'll talk about later. Trypsin is a very useful enzyme that is used to cut proteins. All right? Enzymes that cut proteins have a name. They're called proteases. So trypsin is a protease, meaning it cleaves peptide bonds in other proteins. Will it cleave peptide bonds in other trypsins? The answer is yes, it will. So if you leave trypsin in a tube long enough, it'll basically digest itself as well as digesting everything else. Kind of a cool thing. Okay. What trypsin does is it cleaves the bond on the carboxyl side of lysine or arginine. It cleaves, and when I say carboxyl side, what does that mean? Well, here's a lysine or arginine side chain. There's the carboxyl of the lysine, there's the amine of the lysine. It's cutting on the carboxyl side. We can see it's cutting this bond right here. There is that bond that just got cut. Okay? So trypsin cleaves consistently on the carboxyl side of lysine or arginine. Now, not coincidentally, lysine and arginine are two of the amino acids that have R groups that can have a positive charge on their side. They have that extra amine on there. And we'll see later when we talk about a related protein called chymotrypsin that there's a very important reason why it's cleaving specifically at that place. That'll happen in actually probably next week. I'll talk about that. Okay. There are a series of agents that we can use. I'm not going to ask you to memorize this table, but there are some things on here that I think that you should know where they cut. One was cyanogen bromide. I've already talked about that guy there. And cyanogen bromide, as I said, is a chemical reagent. It cleaves on the carboxyl side of methionine. It's very commonly used in laboratories. You can see there are other ones, but I'm not going to expect you to know those. I do think you should know about trypsin, however. Trypsin is very simple. It cuts on the carboxyl side of lysine and arginines. Thrombin cuts on the carboxyl side of arginine. That's a simple one to know. Staphylococcal protease, very simple. Look where it cuts, only on the acid side, the carboxyl side of the acids. And you'll notice these are all cutting on the carboxyl side except for the last one. Now the last one is a very useful one. It used to be actually used in some protein sequencing methods because it was a way of sequentially cleaving amino acids off of the carboxy terminus. Carboxypeptidase A is an enzyme that will start at the carboxyl end of a protein and clip an amino acid off. And then it'll move in and it'll clip another amino acid. And then it'll move in and it'll clip another amino acid. Now, as noted here, it's not very good if that terminal amino acid is arginine, lysine, or proline, but for our purposes, we're not going to memorize that statistic. We're just going to treat carboxypeptidase A as if it can cut off anything off of the carboxy terminus. Why does it cut on the amino side? Well, the carboxy side isn't attached to anything. It's a free carboxyl end. Now, these reagents are very, very useful for us. I'm going to talk later about chymotrypsin. You don't need to know the specificity because chymotrypsin is odd. It's willing to cut at a variety of different amino acids. So it's a little confusing. You can see most of them are aromatic. There's tyrosine, there's tryptophan, there's phenylalanine. Those are all aromatic. You see leucine, that's hydrophobic, and you see methionine, that's also hydrophobic. So there's a sort of a hydrophobic tendency that they have, but I'm not going to expect you to know that. But I will talk more about chymotrypsin when I talk later about mechanisms that enzymes use to cut things. Okay. All right. I'm not going to talk about overlap analysis. I'm not going to talk about disulfides. I'm not going to talk about diagonal electrophoresis. I'm skipping through a lot of things here. Now, what I want to do is get down over here and talk about some methods that are interesting and that are useful for us. I've already mentioned MALDI-TOF, so I think I'll actually start with MALDI-TOF, and then I'm going to come back and talk about Western blotting. So I'm going a little out of sequence here. MALDI-TOF is shown right here. Now, if you've ever taken an organic chemistry laboratory, you've probably learned something about mass spectrometry. If you haven't, I will tell you very briefly. Mass spectrometry is a technique for determining the mass of a molecule. Modern mass spectrometry is exquisitely sensitive and exquisitely able to determine those molecular masses. And part of that wonderful ability that is now available in mass spectrometry is due to this sophisticated mass spectrometry technique called MALDI-TOF. Okay? MALDI-TOF has a long acronym. I'm not going to go through that. But the TOF, the T-O-F part, is called time of flight. And I'm going to, that will be important as we shall see. So how does MALDI-TOF work? Well, a mass spectrometer basically has a vacuum chamber. And in that vacuum, chamber, there are small ions that are ionized forms of the molecule that we're interested in determining the mass of. Because they are ionized, as we will see, the method relies on their having a charge, number one. And the method relies on us having a detector to detect when those ions slam into it. So there's two things we have to have for this mass spectrometer. We have to have an evacuated chamber, three things. I guess an evacuated chamber, we need to have ions, and we need to have a detector. So how does the process work? The process works by our taking a sample. This sample might be, let's say, a small peptide that we've cut up and we've put into the MALDI-TOF device. Okay? So I've gotten my small peptide. I've already broken into a small piece it's small enough that I can work with easily. And I embed that peptide into a crystalline substance. It's a crystalline substance that actually is volatile. It's volatile in the chamber in which this has been placed. There are a variety of compounds that you can use for this. The volatility is such that it can be controlled. All right? And that's a critical part of MALDI-TOF. So we can imagine right here a little sample. It's in a little tiny crystal. Okay? So my material that I'm interested in is embedded in this crystal, and it's waiting for me to volatilize it. All right? So I introduce it into this vacuum chamber, and yes, that takes a little sophisticated technique, but you get it into the vacuum chamber. It's sitting here waiting for the process to get started. The process starts with the activation or the volatilization of that material by a laser. A laser shines onto that little crystal, and the crystal material vaporizes, and that leaves the compound that I'm interested in determining the molecular weight of in the chamber. I'll slow down. It leaves this material that I want to know the molecular weight of in the chamber. This is a vacuum chamber. So there's no air, there's nothing else that's in there. Here's this material that I'm interested in. The loss of the crystalline material left that material ionized. That's an important step. It left that material ionized. That ionization resulted in my material having an excess positive charge. And now you see these little rings right here. These little rings are magnets. They help to focus these ions passing through them. So they're focusing them, number one. And why are the ions passing through there? Well, there is a detector down here that has a strong negative charge that is attracting these guys this direction. Now, if we think about this, when we ionize this material right here, if we make note of the time at which we ionize it, and we watch the time very carefully, the time it takes for an ion to move down here all the way to the other end is actually proportional to the mass of the material. If everything in that chamber has one charge, that is a +1 charge, and I have some materials in here that have a molecular weight of, let's say, 2,000, and I have other ones of 4,000, they have the same charge, they have the same force pulling them towards the other end, all right? But due to inertia, the one that has the mass of 4,000 is going to take twice as long as the one that has the mass of 2,000. It's like the difference between pushing a small Honda Civic down the road and pushing a big SUV. All right? If it's one person pushing them, it's going to take a lot more effort, it's going to take a lot more time to get that big guy pushed than the little one, since the force is the same. All right? So, if we measure the time it takes for the ion to move from the crystal down to the detector, we can very accurately measure its molecular mass. That's the simple idea behind MALDI-TOF. Now, using MALDI-TOF, we can determine the molecular mass of things put into it to within hundredths of a molecular weight for masses up to a few thousand. 1,132.31 grams per mole. Okay? That's really precise. If I know the mass of something like that very carefully, and I know the starting material that comprises it, let's say, well, this is a protein, so the starting material that comprises it has got to be amino acids. and each amino acid has its own distinctive molecular weight, I can ask a computer to say, which amino acids add up to 1032.71? Right? So MALDI-TOF, with MALDI-TOF, I can actually get information about which amino acids are in a given fragment. That's really useful. If I'm careful, and this is a little bit more complicated, but if I'm careful in my analysis, I can actually get the sequence of those amino acids in there. Well, how in the world do I do that? It turns out those molecules, when they ionize, they don't just go to the parent ion. They actually break into pieces. And so the pieces come through. So if I start analyzing the masses of all the pieces are going to differ in size by 1 amino acid each. So if my parent ion is 1032.51, and there's another piece that's 952.72, that's a difference of about 100 something, I can say, what amino acid has the difference in mass of those two? I now know one of the terminal amino acids. And I back up and I say, what's the mass of the next peak and the next peak? So that if I have a very good experiment, I can actually read the sequence of the amino acids by comparing the sequence of each of the smaller and smaller fragments that are produced by the ionization. Now that is extraordinarily powerful. Okay? It is so powerful that using a modern mass spectral analysis like this, a person with a robotic system can determine the sequence of 5,000 or 6,000 proteins in a day. That beats Edmond degradation all to hell. Okay? Now, one of the things I haven't told you is that there's a computer program that interfaces with these that really help us a lot. Okay? If I'm sitting here with a calculator and I'm trying to determine those molecular masses and those differences, that's not going to be very good. All right? If I have a computer that not only has all that protein information but all the genomic sequence information, that can be great. Because now I can not only have this instrument tell me what the individual peptide is, but I can go to the computer and say, well, which protein in the cell has the following sequence? Alanine, alanine, cysteine, lysine, leucine. I might find 2 or 3, but I have all of a sudden narrowed down from 5,000 or 6,000 possible proteins to 3 or 4 possible proteins. If I compare other fragments from that same protein, it's very easy for me to see exactly which protein it is I'm talking about, and the computer can do it without me even having to think about it. So it's a very, very powerful technique. When I talked about proteomics, all right, the other day, and I said that we use 2D gel electrophoresis to do these phenomenal analyses to determine differences between different treatments for what proteins are produced, MALDI-TOF is another important tool for proteomic analysis, because it's with MALDI-TOF that we can actually take a 2D gel and figure out which protein is which spot. That's how we did it. So a robotic method would take a 2D gel, and the little robot would cut out each one of those little spots. It would digest it with proteases to break it into pieces. It would feed it into the mass spec. It would analyze those fragments. It would look at the genome and say, here's the protein here, here's the protein here, here's the protein here, all while you're getting coffee. This is the kind of science you should all go into. This is great work, right? Good work if you can do it. The computer does all the work for you. Okay. Questions about that? Okay. Let's see. I said I would talk about Western blotting. Let's talk a little bit about Western blotting, and then I'm going to move on to talking about hemoglobin. All right. Western blotting is one last technique that combines a couple of things. So we talked about 2D gel electrophoresis, and you saw how I did 2 different things to do that. Western blotting uses another approach to identifying proteins of interest. The reason we do Western blotting is it's fairly simple and it's fairly quick. We don't all have access to a MALDI-TOF to do all of our work. So Western blot is a real good way for us to see, for a particular protein, whether or not it's being made in a given cell. How do I determine that? Well, the way I would determine it is I would take my cell and I would bust it open, just like I've done with all these things. I might do a simple purification, but then I would have a mixture of proteins. The mixture of proteins I run on an SDS page to separate the individual pieces, so I get this banding pattern that are sorted by molecular weight, the largest ones at the top, the smallest ones at the bottom. And I'm interested in a particular protein. I want to know if this particular protein that I've been studying is present in these cells or not. That's what Western blotting tells me. To answer that question, I have to have a way of detecting that protein, and only that protein. Well, it turns out that the way that we specifically detect proteins in the laboratory is by using antibodies. So antibodies are proteins of the immune system that are agents that will bind specifically to only a single protein, or a single structure. It doesn't even have to be a protein, but in this case it's a protein. Okay? Well, how do I get antibodies specific for my protein? Let's say I'm interested in a DNA polymerase protein from beetle. Okay? How do I get an antibody against that protein? Well, in the laboratory it's actually fairly simple. I purify my protein first. I take that purified protein and I squirt it into usually something like a bunny rabbit. Okay? Might be a goat or some other organism. If that protein wasn't naturally made, for example, I wasn't working with, let's say, bunny rabbit DNA polymerase, if it wasn't being made normally by the rabbit's immune system sees it as an invader and starts making antibodies against it. So that's how the antibodies get made. I then simply have to go and take a little bit of the blood from the bunny rabbit, purify away the antibodies, and I've got what I'm after. It doesn't hurt the rabbit, it doesn't kill the rabbit or anything like that. It's fairly straightforward. The rabbit serves as a way for us to have a reagent that we can detect a desired protein. Well, now I've got my antibody in hand. I've got my gel that I've run. What do? Well, first of all, I take that material that's on the gel and I use techniques of electricity to transfer the material from the gel onto a membrane. So basically I put a little electrode on the top and I just blast it with electricity so that it leaves the gel and goes, and the proteins that are there leave the gel and go and stick to the membrane. They will physically stick to it. So I've got my proteins on the membrane. I put the membrane in a baggie, kind of like you see here, that has a buffer, and it has the antibody in it. Well, the antibody's been modified so that it has a colored reagent stuck to it, like it's blue or it's green. Or maybe it has an enzyme that gives a blue or a green color when I treat it with something. It doesn't really matter. But I have some color reagent that allows me to see where the antibody is. I let this mix for a while, and of course the antibody is only going to bind to, presumably, my protein. It's not going to bind to the other proteins that are there. I take it out, I wash it off to get rid of the antibody that's not stuck to the membrane, and if all goes well, what I see is this guy over here. And I look at this and I see a band and I say, yes, in fact, I did have my protein in this mixture. Does that make sense? So the antibody technique of Western blotting is really useful. You might wonder where the name came from. The name is actually a joke. All right? So there's a similar technique that's used to detect DNA fragments. It was the first blotting technique that was invented, and it was invented by a man named Southern. His real name was Southern. So when somebody came along and they invented a technique to detect RNA, they thought it'd be ha ha ha, we'll call it Northern. And then somebody came along and decided they were going to invent a technique, or they did invent a technique to do proteins, and so the logical thing was to call it Western. So they call it a Western blot, and that's why it's a Western. Okay. Questions on that, or can I move forward and talk about hemoglobin? You guys are the quietest class I've ever had. I don't know. If that's good or bad, I hope it's good. Maybe I'm being too clear. I don't know. All right. All right. Hemoglobin, I think we're going to find is, as I said, it's usually one of the most popular lectures I give, and there's a lot of material in hemoglobin, so I'm going to try to get through this today, and I've got quite a bit to do with that. So I've already introduced hemoglobin a little bit. I told you it had 4 polypeptide subunits. It had quaternary structure. I told you it was very good at carrying oxygen and giving up oxygen. I told you that contrasted with myoglobin, which only had a single subunit and was mostly good at holding on to oxygen. It's not very good about giving it up. The oxygen-carrying component of both of these proteins is known as heme. And you see on the screen a depiction of what heme looks like. We're not going to draw the structure, obviously, but it has a critical component. First of all, it's what we call a protoporphyrin ring. And porphyrins are large, flat, ring-like molecules like we see here. Another place that we see porphyrins is in chlorophyll. Chlorophyll has a ring structure very, very similar to the ring structure that we see in heme. The difference being that chlorophyll has magnesium there instead of iron. Now, iron is the atom that actually carries the oxygen. It carries molecular oxygen, that is O2. It carries it through an attraction, as we shall see. And keep in mind that this heme is attached to the polypeptides of the individual subunits of hemoglobin or the single subunit of myoglobin. So somebody asked, I think it was Anesia the other day, is it physically attached? The answer is yes. It's covalently physically attached to histidines. There's a histidine that attaches it over here, back over here, and back over here. You don't need to know that, but that's where it gets attached. Okay, now, heme turns out to be not only critical for carrying things, but also for giving the hemoglobin and the myoglobin some of its properties. So let's take a look at oxygen binding first of all, of the heme group. Okay? Now, your book's a little odd. It goes back and forth between hemoglobin and myoglobin. The basic thing that heme does is the same in both. Okay? So what you see here for hemoglobin is the same as it is for myoglobin. Now, what you see on the screen is something that is very, very subtle, and it becomes very, very important as we talk about it later. The very subtle difference between the figure on the left and the figure on the right, besides the fact that oxygen is binding, is that this ring starts out being a little bit concave. You see how the iron is slightly below the plane of the ring over here, and you see how over here it has been elevated by a tiny fraction of an angstrom. That movement right there, 0.4 angstroms, that 0.4 angstrom movement is why you are alive. Life as an animal would not be possible without that movement. Now, I hope to convince you of why that's the case in just a little bit. That 0.4 angstrom movement is what allows you to be an animal and not a bacterium or a plant. Okay? Now, what's causing that movement? Oxygen. Oxygen is attracting, it's literally pulling up that iron atom. Now, this process happens in myoglobin. It also happens in hemoglobin. In hemoglobin, it has a much more drastic effect, as we shall see. I want you to keep that movement in mind. I will come back and talk about it in just a bit. Okay, we don't need to worry about that. All right. Now, if we look at deoxyhemoglobin, okay, we see that deoxyhemoglobin has a schematic structure that looks like this ribbon diagram, and in the space filling it looks a lot more complicated over here. But I'll remind you we have 2 identical units called alpha and 2 other identical subunits called beta. I guess I've got them backwards per what they've got. Beta there and then alpha on this direction here. It doesn't really matter. They're very, very similar to each other, and we don't distinguish their functions because the functions are essentially the same. In fact, they're not only essentially, they are the same. But what I want you to see in this figure is something I'm going to come back and talk about later in the period today, and that is this little donut hole right here. This donut hole, like so many things in hemoglobin, has an extreme importance for human health and also for fetal health, as we shall see. Okay. Now, I'm building up with a lot of big stuff about hemoglobin, so I better deliver on this, is all I can say. Let's look at what happens when we mix oxygen with myoglobin. Okay? What you see on the screen is a graph that plots what percentage, and this in terms of fraction, what percentage or fraction of the myoglobin has oxygen in it on the y-axis compared to the concentration of oxygen on the x-axis. What you can see very clearly as the oxygen concentration goes up, starting from 0, a very tiny amount of oxygen causes this to go boing. It goes up very quickly. What this tells us, first of all, is that myoglobin has tremendous affinity for oxygen. It really likes to grab it, and I've already told you that, so you probably say, okay, that's fine, that's what you told us. Now, myoglobin, if it were all it did was hold oxygen, it wouldn't do us any good because it's holding oxygen for a purpose. It's holding oxygen to give us oxygen in our muscle cells when they really need it. And they have to really, really need it because it only gives it up significantly when the oxygen concentration gets very, very low. But it is kind of like that backup battery that your computer might have when you run out of electricity. You want to have something to keep the juice going so you don't lose the whatever it is that you have stored in memory. And in fact, that's the function that myoglobin has with respect to oxygen. It's like what I like to call an oxygen battery. Okay? Well, that's really great if all we want to do is give up oxygen only when cells desperately need it. Our body needs oxygen all the time. We don't want a protein that only gives up oxygen when we desperately need it. We want a protein that grabs oxygen when we want it to and give it up more readily when our cells need it. And hemoglobin fits that model very, very well. This shows the binding affinity for hemoglobin for oxygen compared to myoglobin over the same concentration range. We notice a big difference. All right? First of all, this curve that we saw for myoglobin is what's called hyperbolic. Okay? It forms a hyperbola. The binding curve for hemoglobin is what we call sigmoidal. And we call it sigmoidal because it has an S shape. And though it's not real apparent here, it is in reality more like a real live S shape. This figure illustrates a very important thing. Okay? It tells us that where the oxygen concentration in the body is very, very high, hemoglobin will essentially have the same amount of oxygen bound to it as myoglobin does. Well, that's good. In the lungs, we want hemoglobin to have high affinity for oxygen. We want it to grab oxygen and be full of oxygen in the lungs because it's going to take that oxygen from the lungs out to the tissues. As hemoglobin leaves the lungs, the oxygen concentration falls off, and when it falls off, what happens to the percentage of hemoglobin full of oxygen? It drops. It drops because hemoglobin is giving up oxygen. Notice that right here, hemoglobin, if we go to this point right here, we've got 25 torrs of oxygen. We've got about 50% of hemoglobin has given up all of its oxygen. Myoglobin is still playing the selfish older child and is holding on to it. Okay? The body's needing some stuff right here. You're sitting here right now. Your tissues are at that level. They want that oxygen. What if they weren't getting that oxygen? They would be in trouble. When we get all the way down to the bottom, only then does myoglobin start to give up the same amount that hemoglobin has been giving up all the way along. Now, why does hemoglobin look like this and myoglobin looks the way that it does? Okay? That's something I think I'll take up after we take a couple-minute break, and we'll talk about a phenomenon called cooperativity. Okay? So let's take a brief break, and we'll come back and deal with that. Excuse me. Sorry. Squeeze back through here, Anesia. I have to tell myself where the break is, so I don't have to